Hi everybody, and welcome to our module on DNA structure. In this module, we'll review some basic concepts of DNA that you should all be familiar with, and basically the point of this module is to set the stage so we can talk about the biochemical synthesis of purines and pyrimidines. So DNA, as you know, contains our genetic code, and it's found in the nucleus of eukaryotic cells. It's found in the cytoplasm of prokaryotic cells like bacteria. DNA, as you all should know, has two complementary strands, and I'm drawing a line through the two strands on this picture of DNA on the screen. Each strand is made up of a series of building blocks called a nucleotide. This is a nucleotide right here. And a nucleotide contains three elements. First of all, it has a phosphate group. Second of all, it has a ribose sugar, which is right here. And third, it contains a base. And these are called nitrogenous bases because they all contain nitrogen derived from amino acids. And there are four bases found in DNA: adenine and thymine, and guanine and cytosine. And the guanine and cytosine are complementary to each other, and so are the adenine and thymine. So part of what can be confusing as a medical student is the vocabulary people use when they talk about DNA, especially when you read textbooks. So what I'm going to do in the next few slides is just define some terms. We need to know the difference between a nucleotide and nucleoside. We need to know what the nitrogenous bases are, and we need to know the difference between purines and pyrimidines. So DNA, as I showed you before, is a polymer. It's made up of repeating units, and those monomer units are called nucleotides. And like I told you before, nucleotides have three elements. They have a pentose sugar, which is this unit right here that I'm circling. They have a nitrogenous base. The base is not shown in terms of its chemical structure, but it would sit right here. And then finally, they have a phosphate group. And if you see an OH group right here, then it is a ribonucleotide. If that OH group is removed, as I've shown you over here, then it is a deoxyribonucleotide because the oxy group has been removed. So usually people say that DNA is made up of nucleotides, and when they say this, they mean it's made up of repeating structures of a nitrogenous base, a sugar group, and a phosphate group. But sometimes you will hear people use the term nucleoside, and a nucleoside is the base and sugar with no phosphate group attached. So if we look at this picture on the right side of the screen, you can see that there is a phosphate group attached right here. Therefore, this is a nucleotide. Specifically, it's adenosine monophosphate, and it's mono because there's only one phosphate group. If the phosphate group were removed, this would be an adenosine nucleoside, and you just need to know that terminology so you can understand what you're reading in textbooks. There are five nitrogenous bases that are found in nucleotides in the human body. Three of them have one ring, and those three with one ring are called pyrimidines. And the three pyrimidines are cytosine and thymine and uracil. And you may know that uracil is found only in RNA but not in DNA. Thymine, on the other hand, is found in DNA but not in RNA. Then there are two additional nitrogenous bases that have two rings, and those are called the purines. And the two purines are adenine and guanine. Once a nitrogenous base is attached to a ribose sugar and a phosphate group, it becomes a nucleotide, and the name of the nucleotide depends on the name of the base that's attached. So when cytosine is the base attached, it's called cytidine, and when thymine is attached, it's called thymidine, and when uracil is attached, it's called uridine, and all the pyrimidine nucleotides end in dine. For a purine, once the base is attached, the nucleotide name ends in sine. So when adenine is attached, it's called adenosine, and when guanine is attached, it's called guanosine. All of the nucleotides are synthesized as monophosphates, so this means they have a base like I've shown here and a ribose sugar, and they have just one phosphate group. They are then converted to a triphosphate form, and the reason this is done is because the triphosphate form is the form that can be incorporated into DNA. You need to attach this phosphate group right here to a hydroxyl group on another DNA monomer, on another nucleotide. In order to do that, you need three phosphate groups right here. This makes the reaction favorable so that these two can be cleaved off, and this one right here can attach to the hydroxyl group on the next DNA nucleotide. There is a phenomena in DNA that you should be familiar with called DNA methylation. When this occurs, a methyl group is added to the base cytosine. So if you look at the bottom of the screen here, on the left side is the structure of the base cytosine. On the right side of the screen, I've shown 5-methylcytosine, and it has a methyl group added right here, and that's called DNA methylation. It's done to the cytosine base. And this occurs in segments of DNA where there are repeating CG patterns. These are called CG islands. And what will happen is the cytosines will be methylated on both strands. So for example, if you have a C with a complementary G here, and then you next have a G with a complementary C here, the cytosine on both strands will become methylated in these CG islands. And when this occurs, transcription of the DNA will become inactivated. This is part of a bigger subject called epigenetics. Epigenetics is the study of the way our genes are turned on or off. So you can have the same genes as someone else, but another person may have them turned on and you may have them turned off. That's the result of different epigenetics between you and another person. Human DNA is about 70% methylated. And the important thing about this to know is that if the body finds lots of unmethylated CG strands floating around, that can stimulate an immune response because the innate immune system will think that all this unmethylated DNA is probably coming from bacteria, and this can trigger an inflammatory reaction. Now, although bacterial DNA has less methylation than eukaryotic DNA, there is some methylation that occurs in the DNA of bacteria. Bacteria can methylate both cytosine and adenine, so this makes them different from eukaryotes who methylate predominantly just cytosine. In addition, methylation serves a different purpose in bacteria than it does in humans. In bacteria, methylating DNA protects the bacteria from viruses, which you may know are called bacteriophages. So these phages like to insert their DNA into the bacteria, but the bacteria will recognize that that DNA is not methylated and they will destroy it using enzymes called endonucleases. This is part of a broader area of bacterial research called restriction modification systems, and these are systems that bacteria use to prevent their DNA from becoming modified or restricted. But the point here to know is that bacteria can methylate their DNA, and they do this to protect themselves from infection with viruses. Now that we've discussed the molecular structure of DNA, what we're going to do for the remainder of this module is talk about how DNA is packaged so that it will fit in the nucleus. The DNA that the human body uses is so large that if it were laid out end to end, it wouldn't fit inside the nucleus. The way it fits in the nucleus is by condensing it in a very specific pattern. So the first thing we need to talk about is chromatin. And chromatin is a structure found in the nucleus of eukaryotic cells, and chromatin consists of DNA plus proteins. And you may be familiar with structures called chromosomes, and chromosomes are units of condensed chromatin. One of the most important proteins found inside of chromatin are the histones, and inside of chromatin you can find repeating units of histones plus DNA, which are called nucleosomes. This is a picture of a nucleosome on the screen right here, and there are many of these found inside chromatin, and this is the way that the DNA condenses so that it can fit inside of the nucleus. So the histones are proteins or peptides, and they go by names like H1, H2A, H2B, H3, and H4. All of the histones contain basic amino acids, and in particular, they have a very high content of two amino acids, lysine and arginine. Lysine and arginine are both positively charged, and this makes them very good for binding to the negatively charged phosphate units on the backbone of DNA. In a nucleosome, you will find DNA wrapped twice around four of the histones. So if you look at this picture on the top of the screen here, you can see that the DNA represented by this red strand is wrapped around four circular histones, H2A, H2B, H3, and H4. It wraps around these units twice so that there is an 8 histone core made up of 2 copies of each of these histones at the top of the screen here. There's another histone called H1 which sits outside of the nucleosome, and it is distinct from the others. It's not in the nucleosome core, it's larger and it's more basic, and it's important for tying together the beads on the string. So these octamers of histones right here are often called beads, and they're tied together like beads on a string by the H1 histones. This is easier to understand with a picture, so you know that DNA forms a double helix, and then DNA can wrap around histones to form a nucleosome, and then you can have a series of those nucleosomes which look like beads on a string, and then the H1 histones will pull all the beads on the string together to make condensed chromatin, and this is nice and tightly packaged, and it will allow the very large human DNA to fit inside of the nucleus. The place where you will most often discuss histones in a clinical context has to do with drug-induced lupus. So drug-induced lupus is fever and joint pains and a rash, very similar to traditional lupus, but in this case it begins after starting a drug. And in patients who develop drug-induced lupus, they will have anti-histone antibodies about 95% of the time. Contrast this with patients who have classic lupus, they typically have anti-double-stranded DNA antibodies. And you can see how these two disorders would look similar because they both involve autoantibodies against DNA. In the case of drug-induced lupus, it's against the histone molecules. In the case of traditional lupus, it's against the double-stranded DNA itself. And there are 3 classic drugs you should know for step 1 that can cause a drug-induced lupus syndrome. And these are hydralazine, which is a blood pressure drug, procainamide, which is an antiarrhythmic, and isoniazid, which is a tuberculosis drug. Chromatin can exist in 2 forms. The first type is called heterochromatin. This is very tightly condensed chromatin, and when chromatin is like this, the gene sequences will not be transcribed. And this varies by cell. So, for example, cardiac cells may transcribe some genes that kidney cells do not, and vice versa. And remember that I told you before that DNA methylation tends to inhibit transcription, and you will find a lot of DNA methylation in condensed heterochromatin. The other type of chromatin is called euchromatin. This is less condensed. Transcription can be active in euchromatin, and there is a significant amount of acetylation of histone going on in euchromatin, which is one of the things that allows the genes to get turned on. So let's talk about histone acetylation. Acetylation means adding an acetyl group to a molecule. In the case of histone acetylation, we are going to add an acetyl group to lysine. Recall that I told you that histones contain a lot of lysine amino acids. This is the chemical structure of lysine here. This is what an acetyl group looks like, so we are going to add this acetyl group to lysine. And when we do that, the chromatin will relax and transcription can occur. If you look at this picture on the bottom of the screen here, we have some closed chromatin on the left side of the screen. There are enzymes called histone acetyltransferases which will add acetyl groups to lysine. This will relax the chromatin and allow transcription to occur. There is also a process called histone deacetylation, and this has the reverse effect. It packs the chromatin tightly together and it blocks transcription. So on this slide, I've summarized the two epigenetic phenomena that I think it's important to know for step one in a way that will hopefully stick in your mind. So if you have DNA that wants to be transcribed, if you acetylate the histones, that will allow transcription to occur. On the other hand, if the DNA, especially the cytosine in that DNA, gets methylated, then transcription will not occur. And these are two epigenetic phenomena. There are many, many more. The field of epigenetics is very large and diverse. There are drugs in development called histone deacetylase inhibitors that have potential therapeutic effects. You may one day see them used as anti-cancer drugs. There are some cancers that have increased expression of histone deacetylase enzymes, and so if you inhibit those, it's thought that you may limit growth of the tumor. Huntington's disease is also a disorder where histone acetylation may play a role. I discussed this in the neurology modules. It's a movement disorder where patients have an abnormal protein called the Huntington protein. This is a gain of function disease. There's a mutant protein that is overactive. And one of the possible mechanisms by which this Huntington protein becomes overactive is through histone deacetylation, which silences genes that should control the Huntington protein. And it's thought that this may lead to neuron death in the striatum. So there are histone deacetylase drugs being studied for treatment of Huntington's disease. There's a seizure drug called valproic acid, which has some histone deacetylase inhibition activity. There's a nice review article down here on the bottom of the screen if you want to read more about these drugs that are in development. And that concludes our module on DNA structure.